I think she's been putting all the new stuff in caps, like besides the interview, but like this is the same, that's new, that's different. Like she's been putting the different stuff in caps, which is helpful.
Okay, let's do this. We will start this show. Everybody ready for a podcast?
Yeah.
Yay! Let's have a podcast. Starting in 3, 2. This is TWIS. This Week in Science, episode number 760, recorded Saturday, February 15th, 2020. How to make a protein. I'm Dr. Kiki, and today we will fill your head with thoughts and questions. But first,
disclaimer, disclaimer, disclaimer. From the beginning of human civilization, nature has been altered. The first organic farm was a shock to the senses. What are these strange grains are in this field? It looks like the grains from the far side of the valley and also the ones from the other side of the mountain. What strange goats are these? They're smaller than normal goats, and they're not afraid of people. What strange fruits you have growing on the vines? They're so much bigger than the ones I normally can pluck. And what is this insane claim you are making about owning things? These goats, that grain, those fruits, you haven't even plucked them. How can they be yours? And while humans have altered nature in greater ways since the invention of farming, it is important to remember that altering nature has always been an essential strategy for our survival. For example, the hand-picked story you are about to hear on This Week in Science, coming up next. This is the part where the audience dances.
if it's loud enough and you can hear it, then we dance.
Discoveries that happen every day of the week. There's only one place to go to find the knowledge you seek. I want to know
What's happening? What's happening? What's happening?
this week in science.
Yeah.
What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and our listener out there. Welcome to another episode of This Week in Science. We are live, recording live at the annual American Association for the Advancement of Science Conference in Seattle, Washington, on the SciMike Studio stage, presented by This Study Shows, a Wiley podcast. And we are so glad to be here. Thank you, everyone in the audience, for joining us today. Welcome. Do you like science?
Yeah.
Yay, I hope
We got the good crowd.
we got it.
start.
It's a good start. All right. On this week's show, we have a little bit of discussion about the science here at the conference, and we have an interview. But as we jump in, I would like to remind you that if your friends aren't subscribed to This Week in Science, maybe you should tell them to subscribe. And you can send them to twis.org to find information, or just tell them to search any podcast directory, YouTube, Facebook, all of the things. Look for This Week in Science. They'll find us. Subscribe. Okay, I would love to introduce our guest. Introduce our guest. Dr. David Baker is professor of biochemistry and the director of the Institute for Protein Design at the University of Washington. And he is here at the AAAS meeting talking about his work in designing proteins from scratch. I would love to know, why do we want to do that?
Well, proteins are the miniature machines that carry out essentially all the important functions in our bodies and in all living organisms. And the proteins that we and everything, all other life forms have in them, are the products of millions of years of evolution. And they evolved to solve the problems that came about during that time, like running away from tigers and digesting food and things. But we face a whole bunch of new problems today. The planet's warming up. We're producing all these toxic compounds, and so we know how powerful proteins are from all the examples we have in nature. But those proteins don't solve the current problems. So basically, the reason why we're designing brand new proteins is to solve the problems that humans have created that didn't really arise during evolution.
Why didn't they arise during evolution? I mean, when we think about evolution, it's these modifications through time to solve problems and to allow an organism to survive better, more efficiently. So don't you think that the things that we need would be here by now? Why do we need to keep going?
One of the big problems we face is huge mounds of plastic in the middle of the ocean.
Yeah.
And those were not there 50 years ago. And evolution takes millions or if not billions of years. Another example is Alzheimer's is a big problem now. It wasn't a problem 200 years ago because people didn't live long enough. So
we just died.
Yeah, we just died. So there just are a lot of new problems.
And I guess some of it might be that that protein that we're looking for could already be out there, but finding it is the trick. It could exist somewhere in nature, but we would have to look through every protein everywhere in nature to find something that we want for this specific task versus just.
Well, it's useful in that regard to think about some numbers. So the number of possible proteins. Proteins are made out of amino acids. There's 20 types of amino acids, and typical protein would be a string of 100 or 200 amino acids. Even for 100 residue protein, the number of possibilities is a number. It's 20 to the 100th power, which is 10 to the 130th power. All proteins. You took all the proteins that existed in nature and have ever existed. It's a tiny, tiny, tiny fraction of that. It's number like 10 to the 20th. So it's like the tiniest grain of sand. So there's a good chance the proteins we need don't exist in nature, and that's why we have to make them.
And even then, wouldn't it need to be folded in the correct way to actually have? Okay,
that's right.
There's a whole other layer on top of it.
Exactly. Yeah.
You mentioned that you were talking about folded yesterday, and we have talked about folded for years on the show, and appreciate the way that this crowdsourced human ability to find patterns has enabled us to start really digging into how proteins fold and discover those first principles of that conformation change, how it goes from a single strand of amino acids into folding and connecting and then tertiary forms and all these changes. So how do we really start? How do you start approaching this problem of a new protein when you're like, I've got a sequence of DNA, I've got a sequence of amino acids. What's it going to look like? How do you go between those steps?
Well, there's two separate problems we're working on and that folded players have approached. One is, here's a new gene from some new organism that lives somewhere on Earth, and here's its amino acid sequence, and fold it up and tell me what it does. And that's called protein folding problem or protein structure prediction problem. What we are now doing more of in both my group and on Folded is the opposite problem. Let's say we have a problem. We want to break down plastic or we want to cure cancer, and there isn't a protein that already exists that does that. So let's make a protein which does that, which could be an improved cancer drug. And so we first think, well, what properties would that protein have? And we make a computer model of that protein, and then we design an amino acid sequence which will fold up to that protein structure, and then we make a synthetic gene. This is a brand new protein. We can't. There's no gene that already exists in nature. So we make a synthetic gene that encodes that brand new protein, and then we put that into a bacterium, and the bacterium makes the protein, and then we can get the protein out and see if it cures cancer.
What does it do? So how do you know how the body or a cell is going to react to these proteins?
Well, we've designed them to interact with the body in a very specific way. For example, to suppress an autoimmune reaction or to kill tumor cells. So that's the way we designed it to work. And we know before we actually would get to trying it out in a person that it actually does that sort of sort of biochemically. But there's also the possibility it would do other things, which would not be good things. And for that, that's a risk really with any new type of drug. So there'll be clinical trials starting on the first design proteins we made later this year, and then we'll learn.
Try it out and see what happens. That's amazing.
And this is proteins have applications in absolutely everything. I mean, enzymes alone are used in all sorts of industry. They're in everything that we do. How are you narrowing down where to start?
That's a really good question. That's almost the hardest question of all. So to amplify what you said, some of the things that proteins do is they're involved in forming these really cool materials like tooth and bone and abalone shell. And we're trying to design new proteins that will get inorganic chemicals to grow and crystals to grow out of them, but out of semiconductors instead. So imagine you could make brand new materials that the world has never seen before. And then, of course, we have disease, trying to cure disease. We have a big effort now on vaccines, trying to make a universal flu vaccine.
Yeah.
We're working hard on coronavirus now. So there are a lot of different things. So one good thing is there are a lot of people excited about this area who come to the UW now to try and design new proteins. But figuring out exactly what the best, most important thing to work on at any given time is, we're always looking for ideas.
Is this something that you'd want to then scale to be able to send this ability to make brand new proteins to other universities or to people's garages, or is that something that you would want help with, or is there a reason that you want to keep it kind of controlled for right now?
Well, no, we really want to get it happening everywhere. So Hold It is an effort to get the whole, the general public involved in designing proteins. And in fact, we published a paper a couple months ago showing that folded players can make brand new proteins. It was a totally unsolved problem just a few years ago, but now we've got it so folded players can make totally new proteins. And many people who have been in my research group go on to other universities and start their own groups, also designing new proteins.
Do you think that, from Blair's question though, do you think that the potential of this, like right now, DIY bio is growing, and people are doing CRISPR in their garage and meddling with biology? Is this the kind of thing that people would be able to do in their homes, or is this still the technology is too large for that to happen at this point?
I think that the computer part, I think people could certainly do in their garage, and then they would need what's needed to actually make the proteins is nothing more than you would have in an ordinary laboratory. So you need to be able to get genes, you need to put them into bacteria and grow the bacteria. So it is something that would be accessible. It'd be a pretty sophisticated garage.
Right. It's a garage with venture capital funding.
Yeah. Well, I don't know if it needs venture capital funding, but someone who is serious about it.
Yeah. What is your main focus and your hope? Like, if this really goes to fruition and you see it go where you dream it to go, what is that?
Oh, we're going to make the world a better place.
Better place through synthetic biology.
Well, there's, like I said, new medicines, new vaccines, new materials, like I mentioned, new types of semiconductors, degrading toxic compounds in the environment. You mentioned catalysts. We're working hard on making new catalysts. So there's no shortage of problems that the human space today that in principle could be solved by proteins.
It's wonderful.
Is there something we have to be careful of when we're designing new proteins? Are there kind of warning signs you need to look out for where this could get out of hand?
Yeah, it's a good question. Well, I think the example of coronavirus it helps think about that. I mean, the viruses have evolved over hundreds of millions of years to be super sophisticated at infecting people, making them sick, and spreading. And there's so many complicated functions involved in that. I think even a bad actor who understood how to use our methodology would simply not be able to do that. And unfortunately there already are well-known ways of killing lots of people with, for example, the 1918 Spanish flu sequence is just it's devastating. It's devastating, and that's known. So I don't think there's much. I think the risk from new design proteins is tiny compared to that what we have in nature, because that's one thing that nature has perfected. Viruses have wanted to spread since the beginning of life.
Yeah, but this really gets the work that you're doing in designing these proteins really gets down to the mechanisms and how do we create this new tool to help us move forward into that better future that you're envisioning.
Yeah.
So where can people find you and find out more information about the work that you're doing?
Well, let's see. On the internet, there's the Institute for Protein Design at the University of Washington, and we have a lot of information there about what we're doing. There's a TED Talk I gave a few months ago that describes some of the grand challenges we're working on.
Great. So we'll link to that on our website so that people can find that. Thank you so much for your time today. Really appreciate getting to chat with you.
Thank you.
It's wonderful. Thank you. Oh, we got claps from the audience. Yay. Thank you for listening, everyone. So we are closing out the show. We've got about 5 more minutes here to be able to discuss AAAS and the science that we've seen that really has struck a chord with us while we're here. And for me, yesterday, I went to a panel on the circular economy, the carbon economy, and how we will be working as humans to be able to address the grand challenges that we are up against. Researchers from around the world are using sociological techniques from here at UW, are using sociological techniques to figure out how to work with people to get them to be more likely to make changes. What kind of language is important in talking about cap and trade or talking about a carbon tax? What kind of words will make it more likely for people to find that agreeable versus less agreeable? And what kind of setups do you need to make that work? Another researcher from Finland was working on the circular economy, which is really an extension of cradle to grave thinking, where everything from the ecosystem and the resources that are available are taken into account in the economy and move through to how the end of life of products and how people interact with them. And then researchers at Riken are using new methodologies to create new polymers for biodegradable plastics. And it was just fascinating. And I found out that natural rubber cannot be replaced by synthetic rubber because natural rubber is all cis proteins, 100%, and synthetic rubber is a mix of cis and trans, and so it doesn't work. So we have to use natural rubber, and now there's a fungus that's attacking it. So we need our natural rubber. So there are ecological things that we're up against. What did you find yesterday?
Well, I went to an amazing talk about the future of robotic companions. It was very interesting. But the thing that I actually take away from it is that there's always going to be some motivation that drives technology. And a lot of the time, it is not what that technology ends up being used for the majority of the time. So with the internet, we had a lot of different drivers that bring us to the better world that we have today with greater connectivity. So yes, there might be robotic companions that are driving the economy and innovation with robotics, but there's a great opportunity for us to have caregivers and better use of robotics in our homes every day. So that was very interesting. I also found out that I might someday be able to have a shirt that is linked via Bluetooth to my phone so I can change the color through an app on my phone, which sounds awesome.
Yeah, the point that you brought from that AI and robotics session that you talked about last night was the fact that the sex industry and sex robots may be leading the drive for these home companions. The technology that will one day be taking care of our elderly could have come from the sex
may
industry.
have a dual use.
It may. Fascinating.
Yeah.
Justin,
So having slaved over fermenters to at random hopefully get a protein that can do something that we wanted it to do. This is my favorite subject at AAAS. For sure.
Have you found anything, Dr. Baker, that has piqued your interest?
Let's see, there's been a lot of really interesting things. I've been mainly involved in sessions where I've been speaking, which to me actually my talks are rather boring. I've heard them before, so.
Not as interesting, yeah, but very interesting to us. So thank you so much for joining us. Did you have anything to add?
My
there?
favorite part at these conferences always is eavesdropping on other people's conversations in the expo hall, outside of sessions. That's where a lot of the really interesting conclusions come from. And I think that is something that I always value in times like this, because people are coming together and mixing together in ways that we don't often in our day-to-day. When we have all of science under one roof, then we get to have conversations across fields that are totally new and different. And I think that's absolutely my favorite part of this conference.
Yeah, so, like the little ear dropping thing that happens is like, oh yeah, good luck with your fungus.
This is the path to scientific recombination. Thank you, everyone, for listening. Thank you for joining us today, Dr. Baker. I hope that everyone enjoyed the show. Let us know. Share it with a friend. I would love to give shout outs to people who help with our podcast, Fada, for your help with the social media, getting the word out, and for show descriptions, Identity 4. Thank you for recording audio for our podcast. And Gord McLeod, thank you for manning the chat room. Thank you also to the Burroughs Welcome Fund and our Patreon sponsors for their generous support. I also want to say thank you to This Study Shows, a Wiley podcast, for supporting the SciMike Studio stage. If anyone is interested in supporting us on Patreon, you can find a link at twist.org or go directly to patreon.com/thisweekinscience. We'll be back on Wednesday live at 8 p.m. Pacific time, broadcasting from our YouTube and Facebook channels and from twist.org/live.
We are also available as a podcast. You can just, I think we should put Bing, This Week in Science, anywhere where podcasts are found, and we should pop right up.
For more information on anything you've heard here today, you can find links in our show notes and more information on these stories at twis.org, TWIS.org. And you can also sign up for our newsletter there, where you get to hear a little bit more of our personal musings on science topics.
Newsletter?
We have a newsletter.
Oh, wow, I can't wait
to
to find out. You can also contact us directly, Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Also on the Twitter at TwistScience, at Dr. Kiki, at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, please let us know.
We'll be back here next week, Wednesday, 8 p.m. Pacific time, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is